Welcome to the Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. Before we dive in, please take a moment to like, subscribe, and share this with your friends and relatives. It really helps support the Science Experience. Today, we're taking a deep dive into a really fascinating article by Dudu et al. It's called Cytosolic DNA Crosstalk in Senescence, a new axis of inflammatory signaling. Basically, imagine your cells getting older and, well, mistakenly sounding this constant alarm bell, flooding your body with inflammatory signals. So our mission today is to figure out the surprising culprits, these tiny bits of DNA showing up where they shouldn't, that seem to be driving this inflammation. And, what this paper suggests for maybe promoting healthier aging.
Absolutely. It's fascinating how fundamental processes change as we age. Cellular senescence, it's essentially when a cell puts on the brakes permanently, it stops dividing, but it doesn't die. Now, initially, that's actually good. It helps stop tumors, helps heal wounds. But the problem starts when these senescent cells just hang around in our tissues long term as we get older. They become, frankly, quite damaging. And a key thing they do is release this complex mix of inflammatory stuff. It's called the Senescence-Associated Secretory Phenotype, or SASP. Think of it like a chemical distress signal. Cytokines, enzymes, growth factors, all pouring out, driving that low-grade chronic inflammation we see in aging, messing with tissues, exhausting stem cells, that sort of thing.
Okay, so it's like a cellular smoke alarm that's stuck on constantly causing issues. And this paper digs into what's actually, triggering that alarm. It seems it's not what you might first think.
Yeah, recent work shows these senescent cells collect different types of DNA outside the nucleus, just floating in the main cell body, the cytosol. And that seems to be the driver for this SASP. Exactly right. And the paper focuses on three main kinds of this misplaced DNA. First, you've got mitochondrial DNA, mtDNA. Mitochondria are the cell's power plants, right? But if they get damaged or leaky, their DNA can actually spill out into the cytosol. It's a damage signal. Second, there are things called cytoplasmic chromatin fragments, or CCFs. These are literally bits of DNA from the nucleus, the main command center. They seem to break off maybe due to damage or stress after the cell stops dividing, and they just escape into the cytoplasm. And a third type, it's retrotransposon-derived cDNA. These are copies made from jumping genes, bits of DNA called retrotransposons. In senescent cells, these genes can get switched back on. They make DNA copies of themselves, and these copies end up in the cytoplasm too.
Wow, okay. So different origins, mitochondria, nucleus, these jumping genes, but they all end up as DNA floating in the cytosol. Is there a common way the cell reacts to them? Like, how does this turn into inflammatory SASP?
Yes, that's the crucial link. All these different types of cytosolic DNA basically triggered the same alarm system inside the cell. It's called the cGAS-STING pathway. Think of cGAS-STING as the cell's internal security system, specifically designed to detect DNA where it shouldn't be. Once it gets activated by this misplaced DNA, it sets off a cascade involving other proteins like TBK1, IRF3, NFB, and that whole sequence is what coordinates the production and release of the SASP factors.
So cGAS-STING is the central hub connecting this weird DNA to the inflammation. Makes sense. But okay, here's where the paper points out something really, really interesting. Bit of a paradox, actually. They found that if you block just one of these DNA sources, say you stop the mitochondrial DNA from leaking, or you stop the chromatin fragments or the retrotransposon copies often, that alone is enough to significantly shut down the entire SASP response. Even though presumably the other types of misplaced DNA are still there, how does that work? Why would stopping just one stop the whole thing?
That is the puzzle, isn't it? And the authors float a couple of ideas, two main hypotheses. First, maybe it's a coordinated sequence. Perhaps these different DNA types appear in a specific order. So mtDNA might leak out first, and that process somehow helps the next type, like CCFs, to form or accumulate. If you block the first step, you disrupt the whole chain reaction. The second idea is about synergistic crosstalk. Maybe no single type of DNA is actually potent enough on its own to really ramp up that cGAS-STING pathway and sustain the full SAP. Perhaps you need all of them, or at least a combination, working together to amplify the signal and keep it going strong. They potentiate each other, you could say.
Ah, okay. So either it's a domino effect or they're all needed to make enough noise together. And figuring out which it is or if it's a mix seems pretty important if you want to design, treatments. Which brings up the obvious question. If cGAS-STING is so central, why not just develop drugs to block that pathway directly? Seems simpler.
That's a really important point, and the paper argues against that, actually. Directly inhibiting cGAS-STING is risky. The big concern is broad immunosuppression. Remember, cGAS-STING isn't just about senescence. It's a vital part of our normal immune defense against viruses and bacteria, and it's involved in things like wound healing. If you shut it down completely, especially in older people who might already have weaker immune systems, you could leave them vulnerable to infections or impair healing. Not ideal.
Right. Okay, so it's too blunt an instrument. You'd switch off the fire alarm for the whole building, not just the faulty sensor. So the alternative they propose is more targeted.
Exactly. Much more specific. The idea is, instead of blocking the alarm system itself, why not stop the things triggering the alarm in the first place? So focus on selectively blocking the sources of that cytosolic DNA. For example, drugs that stop mitochondrial DNA release, or prevent those chromatin fragments from forming, or inhibit the enzymes that make the retrotransposon cDNA copies.
And are there actually ways to do that?
Yes, and some are showing promise, according to the review. There are compounds mentioned, like MDM2 inhibitors, that seem to reduce those CCFs. Others, like BAX inhibitors, might help prevent the mtDNA release. And for the retrotransposons, existing drugs like reverse transcriptase inhibitors, think lamivudine, used in HIV therapy, could block their cDNA production. Preclinical studies in animals look encouraging, reducing inflammation, improving health span, and there are even hints from human studies that some existing antiviral therapies might correlate with fewer age-related diseases and slower epigenetic aging. It suggests targeting the source could be viable.
That's really hopeful. So this deep dive kind of peels back the layers, showing how these tiny misplaced DNA fragments inside aging cells could be key drivers of chronic inflammation. And understand that crosstalk might give us much safer, more precise ways to tackle age-related decline.
Precisely. So the thought to leave you with is this. Instead of just dampening the body's general alarm system, what if the path to healthier aging lies in identifying and carefully disarming the specific triggers these misplaced DNA signals before they even set the alarm off? A much more targeted approach.
A really profound thought indeed. Thank you for joining us on this deep dive. Don't forget to like this video and subscribe to the Sand Science Experience for more exciting insights into the world of science.